What is up, party people? We are back with another video of Animal Form and Function Part 6, the immune system. And as always, we're going to be looking at the human or the mammal side of things. Go ahead and pause for today's objectives. All right, let's get into it. So the first thing we need to know is, what is a pathogen? Well, a pathogen is a microorganism such as bacteria, fungi, a parasite, and or a virus that can cause illness or disease within their host, and the host is what they are infecting. So if you get sick, you're the host for a pathogen. What is the immune system? This is what we're looking at. The immune system is a complex of systems of organs, tissues, and cells that defend the body against pathogens and abnormal cells like cancer. There are two types of immunity. You have innate immunity, which is a genetic response. This will always happen. And then you have adaptive immunity or acquired immunity, and this is a specific response. So this is like when you're given a vaccine, which is meant to go ahead and teach your immune system how to handle that specific pathogen or virus or strain of the flu. And then later, once you get it again, then your body is meant to remember that, meaning the adaptive immunity has a memory of the previous encounter, and it should be able to deal with those pathogens quicker and more efficiently. So the innate immune system, it includes physical, chemical, biological, and physiological protections against pathogens. There are non-specific responses, which are considered generic. It's the first line of defense. And our first line of defense is our skin. And our skin provides a physical barrier to prevent pathogens from entering the host. And keep in mind that pathogens have to enter the body some form, some way, typically through openings like your mouth, your nose, a cut. All of those are ways that pathogens can infiltrate your system. Chemicals, the pH of our skin, it's actually slightly acidic, which helps prevent pathogen growth. Our biological is the fact that our bacteria is naturally found on our skin, which then outcompetes the pathogens for resources and can outright kill them. So if a pathogen lands on our skin, not only is the pH going to attack it and cause an environment where it can't thrive, but there's already bacteria that's naturally there that we have a symbiotic relationship with that is going to outcompete it for resources. And then finally, our physiological, sweating, or maybe not sweating, like another excretory gland. The skin adjusts to the environment, which will impact the pathogen's ability to survive. In addition to the skin, the body has numerous ways of responding in general to any pathogen. One, our nasal cavity. We have hairs that block pathogens from entering through the respiratory systems. The mucus, it traps pathogens for removal. The salty environment in our eyes, it's damaging to pathogenic cells. Low pH of gastrointestinal tract, it kills the pathogens, again, because it can't live in that low pH environment. Our osmotic regulation, it results in the flushing of pathogens from the body. This is urination. Our tears, as I mentioned before, they also flush pathogens out of the body. And then we also have various blood and tissue components, which we'll get into in a little bit. The innate immune system response begins with pathogen recognition. We have to know what we're fighting. This is the process of identifying pathogenic agents and or host cells that are already infected. Well, how do we do this? We do this with PAMPs, our pathogen-associated molecular patterns. So these are structures or structural patterns that are found on the exterior of pathogens, and they're expressed by viruses, bacteria, and parasites, and they differ from the host cell, which allows for their recognition as foreign bodies. And they can be composed of carbohydrates, polypeptides, and nucleic acids, and the recognition triggers the innate immune system response. The innate immune system also involves a ton of cell types. We're going to go over a few. It's going to seem like a lot, but trust me, you could get way more in depth in the immune system. Starting off with our monocytes. These are leukocytes, white blood cells, that differentiate into macrophages or dendritic cells in response to inflammation. So they circulate in the blood and lymph, and they help differentiate when it moves to the infected tissue. On the other side, we have macrophages. These are the large phagocytic cells that engulf pathogens, foreign particles, and cancer cells. So remember, phagocytosis, this is where we're engulfing and killing. They're found in the blood, and they migrate into tissues where they're needed. And the activation results in the stimulation and activation of other immune system responses. So the macrophages get there, and then it sends signals to other cells to also come in for backup. And our macrophages also contain pattern recognition receptors, PRRs, and these are molecules on host cells that recognize and bind to the complementary PAMPs. The PAMPs, which we previously discussed as pathogen-associated molecular patterns. Now, I mentioned dendritic cells. Well, what are they? Our dendritic cells, if you remember our previous unit on neurons, they have dendrites. They bind to the PAMPs and they trigger the engulfment and pathogen destruction. They have two or more dendrite-like projections, and their toll-loc receptors, our TLRs, are similar to the PRRs on the dendritic cells that help recognize the common molecular patterns and can distinguish them from the host cells, which is important because we don't want to kill the host cells. We want to kill the foreign cells. And then our neutrophil. These are also phagocytic leukocytes. They're the most common leukocyte, and they're typically the first immune system to arrive once the immune response has been triggered. Their goal is to kill pathogens primarily through phagocytosis, but they also release toxins that kill or inhibit the growth of fungi and bacteria, which is good because we can either kill them or we don't want them to grow anymore so that we can attack the ones that are already here. And then also they will recruit other immune response cells to the infection site, which is important for killing larger pathogens. Next is our eosinophil. These are also phagocytic leukocytes, and they're the recruiters. They recruit other eosinophils to surround the larger pathogens, the bacteria, the fungi, and the parasites, and they'll release toxins that damage and kill the pathogen. But they can also cause host tissue damage, which isn't the best. But keep in mind, the eosinophils are like all of the little minions working together, and they pile on top of this foreign body, and then they all release their toxins and will kill it. But if host cells are around, unfortunately, they'll get caught in the crossfire. Next is our mast cell. It's a leukocyte that produces the inflammatory molecule in response to a large pathogen. Under this, we have a histamine. The histamine is the signaling chemical involved in allergy and hypersensitivity responses. And they're also involved in wound healing. They recruit the macrophages and neutrophils. So again, this is an entire system of telephone and recruiting and trying to kill off those foreign particles. Next is our basophil. They release chemicals that stimulate the additional immune responses, so like allergies, hypersensitivity, inflammatory responses, and defense against parasites. Next is our lymphocytes. Lymphocytes are leukocytes that are involved with the destruction of host cells that are infected with viruses or they're cancerous. Next, you really want to know these, and we'll go over them more in depth later, but natural killer cells, NK cells, T cells, and B cells. We'll go over those in a little bit. Also, our lymphocyte counts are used in the identification of active infection, because if you have a ton of lymphocytes, then we know there's an active infection. Our cytokine release, they are binding of the PRRs and the PAMs, and it triggers a signal cascade that results in the production and release of cytokines. The cytokines are a chemical messenger that regulate the cell differentiation and proliferation, and the gene expression that impact the immune responses. There are 40 different kinds known in humans. And then our interleukins, our ILs, are cytokines that are involved with the communication between the leukocytes, aiding in the communication between the innate and adaptive immune responses. Our interferons, these are also cytokines that are released by the infective cell that communicate their infected status. They can inhibit viral replication and growth of cancer cells. They can also recruit other immune response cells like natural killer cells or macrophages. And then they also have nearby cells that will adjust gene expression to increase the resistance to infection. Now, we've talked about cytokines a lot, but what is a cytokine burst? Well, this is exactly what you think it is. It's like a balloon popping or having just a massive bubble burst like gum. It's a large release of cytokines resulting from the release of cytokines by infected cells that then bind and trigger the cytokine production and are released by the uninfected cells. So again, this is just a massive burst of our immune fighting protection cells. An innate immune response. We've talked about the immune system, but what about the response as well? The initial cytokine release recruits the immune response cells, resulting in inflammation. Inflammation happens because we have a bunch of cells gathering at this one location. So it's a localized swelling, redness, heat, and pain. All of these things are inflammation. It's resulting from the movement of fluid and leukocytes into the infected tissue, which might seem bad. It might be painful. It might hurt, but it's actually good because you know that your immune system has kicked in. Now for phagocytosis, we've been talking about this a little bit, but just to reiterate, it's the majority of innate response, and it involves the cells that engulf and digest pathogens. The larger pathogens require multiple cells to surround the pathogen, break it into smaller parts and pieces prior to phagocytosis, where then it can be engulfed and digested. Natural killer cells. These are the cells that help us identify and kill the infected cells by viruses and tumor cells. The alteration of the MHC1 molecules are resulting from the infection that are identified by the natural killer cells. MHC1 is a major histocompatibility complex. These molecules are proteins that are located on the surface of nucleated cells. And then upon identification of an infected cell, the natural killer cells release periphorin, which is a destructive protein that creates a hole in the target cell's membrane, and granzymes, which are proteases that destroy the target cell proteins, inducing apoptosis. Yes, I said it, apoptosis. I hope you guys remember that from last semester. Go ahead and pause to take a closer look at these different photos that help give you a visual differentiation between the different cell types. Now for a complement system. It is a soluble protein that destroyed the extracellular pathogens, and we have 30 known proteins. The proteins will bind to the surface of pathogens tagged by the antibodies. Once they're bound, the additional proteins will bind, eventually covering the pathogen. Multiple mechanisms result in pathogen destruction. One, the complement proteins work together to break open the pathogen's membrane, causing its cellular contents to leak out. And then opsonization is where the proteins serve as a marker, and they will mark the cell, they will recruit phagocytic cells, and they will enhance phagocytosis. Now moving on to our adaptive or acquired immune system. Well, adaptive immunity is an acquired immunity, which is why they're essentially synonymous. And they develop over time, and it's the result of exposure to specific pathogens. So the exposure is the result of infection or vaccination, which again, we talked about vaccinations are either muted, alive, or dead strains of a particular pathogen, infection, virus, whatever you will. And then you're injected with it, and then your body is meant to fight it off and then remember how it fought it off. So when it encounters it again, it can fight it off easier. But it's all purposeful. And the adaptive immunity, it results in an immunological memory, and it will remember the pathogens, like I just explained, and the system will kick in when the innate immune system cannot handle the infection alone. So if your innate immune system isn't doing so well, like say it encounters the flu, or if you've had COVID-19 multiple times, or if you have a harsh cold, for an example, if your innate immune system just isn't working the best, your adaptive immune system will recognize, hey, we've had this before, let's go back up. And then they will hop on as well. So there's two types of adaptive immune responses. One, there's cell-mediated, which is carried out by T cells. And then there's our humoral, which is carried out by our B cells and antibodies. Antigens. Antigens are a macromolecule that identifies a non-self and triggers the immune system to create antibodies. The antigen-presenting cells digest pathogens into peptides and then display them on their surface through the MHC molecules, which will then be recognized by the T lymphocytes. The antibodies, or immunoglobulin, which is my favorite word from this presentation, can also be known as Ig, are proteins produced by plasma cells, and this is because of stimulation by an antigen. Antibodies have the same structure, except the variable region which determines binding. Our epitopes are the specific portion of an antigen that bind to an antibody. T cells. T cells are lymphocytes that originate in the bone marrow and mature in the thymus. Thymus T cells. They're involved in cell-mediated responses, and they circulate in the blood and identify abnormal cells and foreign antigens. The types of T cells. We have helper T cells, or TH, also known as CD4+. They recognize foreign antigens, trigger the B cell production of antibodies through the cytokine production and release, and then they trigger the macrophages and CTL cells to destroy the pathogens. We have cytotoxic, which is the CTL, or CD8+, which destroy the pathogens, defective cells, and cancerous cells through the release of granules, the peripherin and granzymes. They're activated through the recognition of the MHC1 molecules and T cell receptors. Next is our regulatory, Treg. It's our suppress immune response to prevent excess immune responses. And then finally is our memory, our lymphocytes. It can be a T or a B cell. Both of these cells have memory with the antibodies for specific pathogens. Again, both T cells and B cells have this. It only activates when it's bound to a specific antigen, but it does increase the response time for the immune system activation upon subsequent exposure. B cells are lymphocytes that form and mature in bone marrow. So they both start in bone marrow, but T cells mature in the thymus, B cells mature still in the bone marrow, which is B, bone marrow. They're involved in the humoral immune response, and they produce antibodies that identify and trigger the destruction of specific pathogens. They act as an antigen-presenting cell, so they become memory cells upon the activation, allowing for faster, stronger future immune response. They release the cytokines to help regulate the immune system. And there are different types of B cells. We have plasma cells, which are specialized for high-volume antibody production. These can be short or long-term. And then our naive cells, which are mature B cells that have not yet been activated by encountering their antigen. Then we have regulatory and memory, which is similar to T cells, which I previously explained by the fact that they have memory. Our immunoglobulin classes. These are designated based on their structural heavy chain type, physiochemical, and immunological properties. The two that you most definitely have to know, which were on our Jeopardy game today in class, is immunoglobulin gamma, or IgG, and immunoglobulin mu, which is IgM. Our IgG is the most common, and it provides long-term immunity and protects against bacteria and viruses. It is the only antibody that is able to cross the placenta, which means it's the only one that's able to protect newborns. Our IgM is the first antibody produced during an initial infection, and it's constructed as a pentamer to act as a primary line of defense. We also have IgA, or alpha, which is found in high concentrations in mucous membranes, so saliva, tears, breast milk, and it's the protection of the mucous surfaces from pathogens. So this is important, especially for moms with their newborns, so that they can pass on these immunoglobulins, which is why breastfeeding is so important, especially for newborns. IgE, or Ig epsilon, is responsible for allergic reactions by binding to allergens and triggering mast cells to release histamine. And then finally, IgD, delta. They're found on the surface of B cells, and they help with their maturation and activation. Our antibody functions: 1. Neutralization. They prevent pathogens from binding to their target. Opsonization. They tag the pathogens to recruit phagocytic cells. Complete activation, complement activation. They attach to pathogen surfaces and trigger complement system activation. And then passive immunity. They're temporary immunity provided by the receiving antibodies in blood serum from other individuals. It can be from a blood donation, which can give recipients temporary immunity based on the donor's exposure. And then breastfeeding, like I just mentioned, it gives newborn an increased immunity in the first few months of life. Our antibodies bind with their antigens with varying strength and stability. We have affinity, which is the level of attraction. High affinity is stronger and most stable, and antigens bind fully to the antibody. Avidity is the strength, and stability is the total of the shared binding across multiple antibodies. So multiple antibodies, the strength and stability of them all. And then cross-reactivity is where the antibody binds to the antigen that differs from those that trigger its production. So again, it would be a plus and a minus, not a plus and a plus or a minus and a minus. And then this helps increased immunity by being able to bind to a class of pathogen rather than just a singular specific pathogen. Go ahead and pause to take a closer look at this diagram, but it just shows our rapid response cells, our slow response cells, and the ones that are in the middle also are innate versus adaptive. Here's another image showing the differentiation of non-activated, innate, and adaptive. Immunodeficiencies. An immunodeficiency is the failure or delay or incomplete immune response. It can be acquired or inherited. It can also be influenced by stress, nutrition, deficiency, toxin exposure, radiation, or illness. A hypersensitivity, like an allergen, is an excessive immune response that may cause tissue damage, and it continues after the threat has been resolved, a response to a harmless foreign substance, or a response to a host tissue, like an autoimmunity. So essentially, hypersensitivity is a disorder within yourselves. Your body is interpreting something that should be harmless as something that is harmful. So like pet dander, grass, some more severe allergic reactions like peanuts. All of those are hypersensitivities that shouldn't mean anything to your system, yet for whatever reason, there is a reaction. Autoimmunity is where our antibodies incorrectly mark host cells for the immune response, resulting in tissue damage. This is like arthritis or type 1 diabetes or lupus. Your body is attacking itself. And then allergies is the rapid antibody-mediated immune response to harmless antigens, pollen, pet dander, and food. I hope you learned something from this video today. Make sure you go back over our objectives, and I hope to see you in the next video.